the rest of the things, that is pyrethrum and neem, and the last one is rotenone. These three will be completed today. We'll try to finish it off the biological insecticides today. Okay. Can you all see the screen here?
Yes, ma'am.
Yes. And coming to the pyrethrum, it is a natural insecticide which is derived from dried flowers of chrysanthemum species, and it has been used since ancient time for its potent insecticidal properties with historical records dated back to ancient Persia and beyond. And if you could say about introduction about pyrethrum, pyrethrum refers to the toxic constituents of dried and powdered flower heads of chrysanthemum, as we have studied, and it is one of the oldest organic insecticide. It is used for domestic purpose because of its low toxicity to mammals, non-persistency, and high toxicity to insects, coupled with its remarkable effectiveness in tossing insect knockdown. Okay, and if you could see about the other importance of pyrethrum, Japan was more important as a commercial source before World War II. Okay, and these pyrethrins are esters formed by a combination of two acids that we are going to see in the next slide. These are the chrysanthemum flowers which you are seeing here. And if you could see the composition and the biological activity, the pyrethrins are esters formed by a combination of two acids. If you could see here, this is the structure of pyrethrin. Type pyrethrin 1 and pyrethrin 2. These are the two structures. Whatever the structure which you are seeing here, pyrethrin 1. The esters of chrysanthemic acid are seen here in pyrethrin 1. Okay. And in pyrethrin 2, you are seeing the esters of pyrethric acid. Okay. Not only in pyrethrin 2, you are seeing the esters of pyrethric acid in cynarin 2 as well as in jasmolin 2. In all these three, you are seeing the esters of pyrethric acid. If you could able to compare the structures, you could see the similarity. And in case of pyrethrin 1, you are seeing the esters of chrysanthemic acid. Not only in pyrethrin 1, you are seeing in cynarin 1 and jasmolin 1 too. Okay, all together are called as pyrethrins. Okay, and what all the things which you are seeing here are the chemical structures of pyrethrin 1 and 2 along with cynarin 1, jasmolin 2, cynarin 2, jasmolin 1. Okay, all these 6 compounds together account for the toxicity and knockdown properties of pyrethrum extract. And this is what we have studied with the help of structures. And if you could see the biological activity of the pyrethrum, these pyrethrins act as neurotoxins to insects, affecting their nervous system upon contact. They disrupt sodium ion channels in nerve membranes, leading to hyperexcitation and finally leads to paralysis. Coming to the next aspect. See here, if you could see, this is the chrysanthemum flower which you are seeing. These are the ray florets. And here, these are the disc florets. If you could see here very clearly, you know about the botany, right? This is the corolla, this is the ovary where you are seeing the trichomes. If you could magnify the tritome area, you could see the production of the pyrethrins. This is the place where you are going to see the production of pyrethrins. So this is how in this chrysanthemum plant you are going to see a natural insecticide production called pyrethrin that is used to control the pests or the insects. Okay, these pyrethrins acts on the nervous system of the insects, and that's how it will try to kill the insects to protect the crops. And coming to the mode of action, on direct contact, uh, these pyrethrins affects the insects with their body surfaces. And coming to the neurotoxic effects, upon contact, pyrethrins bind to sodium channels in the nerve cells. If you see the nerve cells, you will see the dendrites, axon, terminals, all these things will be present. Nerves plays a major important role in passing the information to the brain. We have two types of nerves, motor and sensory nerves. Hope you know all these things. So these pyrethrins bind to sodium channels in the nerve cells of insects, causing the prolonged influx of sodium ions, which disturbs nerve function, leading to repetitive nerve impulses, paralysis, and eventually leads to death of the organism. And coming to the next stage of mode of action, this pyrethrum immobilizes insects quickly, making it effective for immediate pest control. These are the things which you are going to see in the insects after it gets subjected with pyrethrum. And coming to the next part, toxicity to insects. How exactly it shows toxicity to insects? Pyrethrum exhibits high toxicity to a wide range of insects, including mosquitoes, flies, beetles, and aphids. Its fast-acting nature and potent neurotoxic effects make it effective against various pest species. Pyrethrum is less likely to induce resistance in insects due to its multi-target mode of action and twig paralysis. If you could see this picture, you're going to see how exactly the pyrethrum disrupts the nervous system in insects. Okay, all the here you are seeing different types of the chemicals and different types of insecticides. Okay, and coming to the next part, toxicity to mammals. Sorry. Hello.
Yes, ma'am.
Yes. Sorry, it got mute. And now, toxicity to mammals. Just now we studied how it is toxic to insects, and coming to the toxicity to mammals, we are going to see here. It shows pyrethrin shows less toxicity to mammals, and it is safe for mammals actually, including humans, when used according to labeled instructions. Yesterday we studied about LD50 and all. Okay, the actual oral, the acute LD50 of pyrethrins to rats have been variously reported, probably for the less pure preparations and 400 to 1500 mg per kilo for the more purified preparations. The acute toxic effects to laboratory animals proceed from excitation to convulsions to paralysis, including muscular fibrillation. Death is caused by respiratory failure. In contrast with human experience, animal tests fail to show the sensitizing effect of pyrethrins. And coming to the expectations, cats are more sensitive to pyrethrum. due to their reduced ability to mobilize, metabolize, and eliminate the compound, potentially leading to toxicity if exposed to high concentration. When compared with the other species, it shows less toxicity to the mammals. And coming to the metabolism, pyrethrum is metabolized primarily in the liver of mammals through enzymatic process. And the metabolites are excreted predominantly through urine with minor elimination through feces. Metabolic breakdown in mammals reduces the toxicity of pyrethrum over time. Coming to the environmental impact, pyrethrum breaks down rapidly in soil and water, reducing its environmental persistence. When used as directed, pyrethrum has minimal impact on non-target organisms such as bees, birds, and aquatic life, and it is considered to have low ecotoxicity, particularly compared to many synthetic insecticides. This is about your pyrethrum, which is a natural insecticide. We all, till now, we have studied about the effective natural insecticide, which is a pyrethrum, and it shows its rapid action. It shows low mammalian toxicity and minimal environmental impact, made it a preferred choice for integrated pest management strategies. So we can use this, but using the limited amount. If it is beyond the limited amount, it may lead to sometimes death of the organism too. Coming to the next part, neem, which is also a natural insecticide, as we all know. Yesterday in our first class, we have covered the importance of several natural insecticides. We have studied the introduction part of insecticides yesterday, where we have covered about pyrethroids. We have covered the first one, neonicotinoids, and we have covered the neem, all these things. And now in the today's class, we are studying about what is the composition, what is its biological activity, what is the mode of action, and how it shows its toxicity to different organisms and all. Okay, coming to the neem, it is a native tree to the Indian subcontinent, and it is known for its medicinal and insecticidal properties. It has been used traditionally for various purposes, including pest control and healthcare. This is the picture of the neem plant, neem tree. And coming to the composition and the biological activity of the neem, we all know that it is a mixture of several compounds. Here, if you could see, it contains several bioactive compounds: azadirachtin, nimbidin, azadiraphol, all these things. Here, these are the two examples which we have given here. These are the two bioactive compounds which you could see in the neem plant. And coming to the azadirachtin, it is a main agent for controlling insects, and it represents 90% effect on most pests. And the content varies from 2 to 4 mg per gram kernel. It acts as a feeding deterrent, growth regulator, repellent, and disturbs the growth and reproduction of pests. Coming to the nimbin and nimbidin, these compounds show antiviral activity. And coming to azadiraphol, it is a feeding inhibitor, causing insects to cease eating in extremely low concentrations. That's how this neem plays a major important role in preventing the insect activity over the crops or the plants. Okay. And coming to the biological activity, these compounds exhibit insecticidal, antifungal, antibacterial, and antiviral properties. If you could see the major compounds which are going to see in the neem plant or neem oil are the things which we have seen: azadirachtin, nimbin, nimbidin, These three already we have studied, and this is meliantrol, quercetin, and salicylic. These are also very important. Salicylic is a powerful feeding inhibitor, and other things which you are seeing here, meliantriol, it is a feeding inhibitor which causes insects to cease eating in extremely low concentrations. And if you could see um, quercetin and all, these also plays a major important role in controlling the insects, and it also acts as a feeding deterrent, growth regulator, repellent, and disrupts the growth and reproduction of pests. So these are the important bioactive compounds which are going to see in the neem plant to control the pests or the insects. Coming to the mode of action, um, you could see many things here. By seeing this, it will be easy for you to understand. This is the picture which is showing the principles of azadirachtin action on insects. Just now we have studied it. Azadirachtin is a bioactive compound seen in the neem plant. It is structurally somewhat similar to different insect hormones, and so the insects do not molt. It also interferes with the function of taste. Hence, many leaf-eating insects are deterred by plants to which even small amounts of azadirachtin have been applied. This is about the azadirachtin, and it shows several things. Here you can see neuroendocrine activity of this azadirachtin and anti-fedency activity of azadirachtin, cellular or molecular effects of azadirachtin, and reproductive effects of azadirachtin. Take an example of anti-fedency. It inhibits this bioactive compound, azadirachtin, inhibits feeding or interfering with chemoreceptors, and it is affecting food conversion or digestive enzymes, and it is affecting the digestibility. This is what we have written here. Antefeedant and growth regulation. Azadirachtin acts as antefeedant, reducing insect feeding and growth by disrupting hormone regulation. Because I already told you that this azadirachtin is structurally similar to insect hormones, eddisones, and seems to be an eddisone blocker so that the insects do not molt. It also interferes with the function of taste. And coming to the next aspect, it also involves in the disruption of metamorphosis. Neem disrupts insect molting and metamorphosis, leading to deformities and reduced viability. Neem oil and extracts repel insects through sensory deterrence. Coming to the neuroendocrine activity, it blocks calcium channel, and coming to the reproductive effects, it reduces the fertility and fertility, and coming to the cellular and molecular effects, it leads to cell apoptosis, disrupting protein synthesis. So all these are the functions of azadirachtin, which is a biocompound which we are seeing in neem plant. Coming to the effect on insects, neem preparations, whether it is a neem powder or neem oil, whatever it may be, the neem preparations show different types of the effects. It leads to the reduction of the lifespan of adults. Insects. We are talking about the effects on insects, and oviposition repellence against the females. It direct ovicidal effects, antefeedant effects against larvae, nymphs, or adults, and formation of permanent larvae. All these things you are going to see in insects. And coming to the toxicity problems, we know that neem products are non-toxic to warm-blooded animals and have been used as a medicine for generations in India. Okay, coming to the exotoxicity and ecotoxicity, it is less harmful than synthetic pesticides. Excessive use of neems can affect beneficial insects like bees and butterflies. Neem products may persist in soil and water, impacting microbial communities and aquatic organisms. Sometimes you may see different aspects with respect to neem. Neem extracts showed no mutagenicity in different types of the molecular biology tests. And the fungus can contaminate the seeds inside the fruits and the aflatoxins, which are extracted and concentrated along with pest control ingredients. Neem oil shows toxicity to certain fish species also. And the seed kernels, whole fruits, or leaves of Melia acidaria contain compounds highly toxic to mammals like toxins. This is about the toxicity problems of the neem. And coming to the end of the insecticide, neem, natural insecticide. Neem offers a natural alternative to synthetic pesticides with broad spectrum activity and minimal environmental impact. So we can use the neem products to control the insects or the pests. This is about the neem and pyrethrum. Hope you understand the things which we discussed under neem and pyrethrum. Under pyrethrum, we have discussed about the composition and biological activity of the pyrethrum, and followed by the toxicity and mode of action. Toxicity towards, towards insects and towards mammals, and we even completed the metabolism. Coming to the neem part, we have completed the composition and biological activity along with the mode of action and effects on insects. By this, we completed the neem and pyrethrum and neonicotinoids. And coming to the next part, we are going to start with retinoids. Give me some time. I'll close this and I'll give you. Hope you all can see the screen, right? Yes. Did you all understand the things which we discussed? Hello?
Yes, ma'am.
Yes. And now we are going to talk about retinoids or rotinan. It is an odorless and colorless.
Which unit is this?
It is the second, actually from the second block. It is from second block, botanicals and conventional insecticides. In the second block 2, unit 5, botanical insecticides. Under botanical insecticides, we are covering or we are studying neonicotinoids,
Okay, now I got it.
neem, rotinan. Okay?
Okay, ma'am. Thank you, ma'am.
Okay. If you have textbooks or if you have any syllabus, you just go through it so that it will be easy for you to understand.
That's why I'm not finding it, so I get confused. Right, ma'am? I opened, ma'am.
Okay.
Okay,
Yesterday,
thank you, ma'am.
Okay. I want to brush it up and I want to give you a hint. Yesterday, we have covered the block 1, fundamentals of insecticide toxicology, where we have studied the formulations and classification of insecticides under unit 1. Hope you remember what we discussed, dust, mites and all. Under classification, we have discussed about organochlorines, organophosphates, carbamates, botanical insecticides and all these things. And under unit 2, we have studied about the evaluation of insecticide toxicity. How much we have to use, lethal dose and all, lethal concentration we discussed. And coming to the unit 3, insecticides and cuticle. How is the cuticle? How the insecticide shows its effect on the cuticle of the insects. And we even studied the effects of insecticides on the nervous system. Maximum number of the insecticides shows its effect on the nervous system of the insects to kill the insects or to inactivate the insects. This is what we covered yesterday along with neonicotinoids, which comes under the unit 5, botanical insecticides. And today we have started with pyrethrum, which is a part of botanical insecticides. Pyrethrum, neem and retinoid. Clear?
Okay, ma'am. Okay, ma'am.
Any doubt?
Okay, ma'am. Yesterday I could not attend the class. Today only I'm attending. I'm following, ma'am.
Okay, then. No problem.
Right, ma'am. Thank you.
Okay. And coming to the last one under biological insecticide, rotenone, which is an odorless and colorless compound. It is crystalline ketonic chemical compound used as a broad spectrum insecticide and also used as a pesticide. It naturally occurs in the seeds and stems of several plants such as Jitama vine plant and the roots of several members of Fabaceae. If you could see the structure of this, it is produced by the extraction from the roots and stems of several tropical and subtropical plant species, especially those belonging to the genera Lontotarpus and Geris. And this is the structure of rotenone. If you could see the structure of rotenone, in 1912, Nade isolated this compound, which having a melting point of 163 degrees from Deris. Just now we have studied that, which was extracted from the different plant species, especially belonging to genera Deris. Okay, and if you could see the molecular formulae, it is 23 C23H22O6, 23 carbons, 22 hydrogens, and 6 oxygen atoms. And the next part here is, if you could see the chemical structure for name of this particular compound, it is hexahydro-2-isopropanol 8,9-dimethoxypromino, chromin 6,1. This is the history about the rotenone. And coming to the mechanism of action, before we enter into the mechanism of action, we'll see few things about the rotenone. Actually, this is used as a fish poison to control populations in reservoirs, lakes, and streams, and to destroy and to eliminate undesired species. Okay, and coming to the other point, it is the most important compound. This rotenone is the most important compound. It has been used in crop protection since 1848. We already know that it is a biological insecticide. And this was extracted from the roots of Deris, which is a species, and also from, as we studied, from other Fabaceae members also. And coming to the mechanism of action, which is very important. Here, it works by interfering with the electron transport chain in mitochondria, because in mitochondria, electron transport chain plays a major important role in the production of energy. To be specific, it inhibits the transfer of electrons from iron sulfur in complex 1 to be ubiquinol. If you could recollect the information which you have studied in your lower classes about the electron transport chain, we have 4 complexes, where all these 4 complexes involves in the releasing transportation of protons according to their gradient. And finally, whatever the protons which gets released or generated involves in the formation of ATP. So this interferes with NADH during the creation of usable cellular energy. Cellular oxygen is reduced to the radical, creating a reactive oxygen species which can damage DNA and other compounds of the mitochondria. Coming to the next part. Coming to the uses of this. It is used as a pesticide, as we already know that. It is also used as an insecticide also, and as a non-selective pesticide. As I already told you, it is used as a fish poison. So It's a non-selective pesticide. It is commercialized as tubes, tuba, or derris in single preparation or in synergistic combination with other insecticides. And rotenone has historically been used by indigenous peoples to catch fish. Typically, rotenone contained in plants in the Fabaceae family of legumes are crushed and introduced into a body of water, and as rotenone interfaces with cellular respiration, the affected fish rise to the surface in an attempt to gulp air where they are more easily caught. It is used in powdered form to treat rabies and head lice on humans and parasitic mites on chickens, livestock, and pet animals. It has been used as an organic pesticide dust for gardens. It kills potato beetles, cucumber beetles, flea beetles, cabbage worms, raspberry, etc. It rapidly biodegrades under warm conditions, so harmful residues are minimal. A light dusting on the leaves of plants will control insects for several days. Coming to the toxicity, it is mildly toxic to humans and other mammals, but extremely toxic to insects and aquatic life, including fish. This higher toxicity in fish and insects is because the lipophilic retinine is easily taken up through the gills or trachea, but not as easily through the skin or the gastrointestinal tract. It is toxic to erythrocytes in vitro. The lowest lethal dose for a child is 143 mg per kg. Human deaths for retinal poisoning are rare because its irritating action causes vomiting. Deliberate ingestion of retinine can be fatal. This compound decomposes when exposed to sunlight and usually has an activity of 6 days in the environment. It oxidizes to retinolone, which is less toxic than retinol. In water, the rate of decomposition depends upon several factors, including temperature, pH, water hardness, and sunlight. The half-life in natural waters ranges from half a day at 24 degrees Celsius to 3.5 days at 0 degrees Celsius. This is all about the uses of retinol. By this, we have completed the complete information of botanical insecticides or biological insecticides. And now we are going to talk about the other types of insecticides, that is the chemical insecticides. Hope you all can see the slide, what I'm sharing here.
Yes, ma'am.
Yes, ma'am.
Yes. Hope you understood the botanical insecticides part. We have covered a total of 4 different botanical insecticides, that is neonicotinoids, pyrethrum, third one is neem, and the fourth one is retinoids. And now we are going to talk about organochlorines. Here in this part, in organochlorines, we are going to talk about what are these, what is its toxicity and its mode of action, and the metabolism of the organochlorine insecticides using examples. And we are going to even study about how exactly these organochlorines show its effect on different species. So that it will be easy for us to understand why we have to use insecticides, why we should not use insecticides. If we are using insecticides, how much amount of the insecticide must be used? All these things will be known to us. And coming to the organochlorines, we already know that these are the insecticides including a group of fluorinated hydrocarbons which comprises DDT and its analogs. These are organic compounds with 5 or more chlorine atoms. If you could see here, you can see the presence of chlorine atoms here in the structure. Okay. And now these organochlorines were the first synthetic organic pesticide to be used in agriculture and in public health. Most of them were widely used as insecticides for the control of a wide range of insects, and they have a long-term residual effect in the environment since they are resistant to most chemical and microbial degradations. Organochlorine insecticides act as nervous system disruptors, leading to convulsions and paralysis of the insect and its eventual death. Some of the commonly used representative examples of organochlorine pesticides are DDT, linden, whatever the examples which we are seeing here. All these are the examples of organochlorine pesticides. If you could see the general properties of organochlorines, these are highly persistent chemicals which are chemically inert and photostable. And these organochlorines are lipophilic, so get absorbed on lipid tissues of living organisms and soil particles. They tend to build up in food chains. They have a high oral toxicity and long residual activity. High persistence makes them ideal for soil application. They have low mammalian toxicity, low vapor pressure, and low aqueous solubility. These organochlorines have low solubility in water. They also have a wide spectrum of activity, tend to kill even natural enemies, leading to the buildup of secondary enemies. Hence, some light DDT have been banned. Some members have both nematicidal and ataricidal effect. Coming to the practical uses of different types of organochlorines. These are the examples of the organochlorines which we have listed towards the left side of the table. And these are the chemicals which controls the pests. This is the mind map or the table for understanding and remembering the things in an easier way. And coming to the mode of action, here they generally destabilize the nervous system. That means these, whatever the organochlorines which we have listed here, all this acts on the nervous system of the insects or the pests. When we see chemically, the DDT is comparatively inert molecule, hence it has the residue problem and the manifestation in food chain. It has a potent broad spectrum insecticide that is also toxic to mammals, fish, and birds. Continuous exposure leads to headache, dizziness, nausea, weakness in lungs. And it also causes different types of problems like blood toxin, and it also causes respiratory problems. It also causes vomitings, etc. When dosages are light, even in the case of frequent or continuous exposure, the pesticides do not often cause visible symptoms of poisoning, but residues tend to accumulate in the body. Coming to the secondary and environmental effects of these organochlorines, these can persist in the soil for months or years of high doses are applied to clay or soil rich in organic matter. Predators which feed on earthworms are threatened because organochlorines tend to get biologically magnified. In plants, organochlorines can be translocated to edible parts. Grazing animals can get the chemical residues in the grass. This ends up in meat, milk, or eggs. And coming to the structure and mode of action of organochlorines, organophosphate, and carbamide group pesticides. This is the organochlorines introduction. Just now we have completed the introduction part of organochlorine, but we have to cover the history. Organochlorine pesticides are chlorinated hydrocarbons which are used extensively from 1940s through 1960s in agriculture and mosquito control. DDT was first commercially produced insecticide in the year 1940, but banned in the US in the year 1970s, but is still manufactured and exported. Coming to the mode of action, there are two types, DDT type and chlorinated type. Organochlorine compounds work on insects by opening the sodium ion channel in the neurons or nerve cells of insects, causing them to fire spontaneously. The insect will go into spasms and eventually die. This is the picture which is representing the normal membrane potential, and when the insect gets exposed with the pesticide, how the membrane potential will be. Here, when it is normal, you are seeing the blockage. Okay, here these are the gates. This is the channel. Okay, sodium channel. Here it is. Here you are seeing the block or the closing of this gate. But because of the exposure to insecticide, you could see the continuous travel of sodium ion into the cell. That's how it caused damage. And these are the structures of different types of organochlorines. In the previous slide also we have seen the same. This is the structure of DDT. And these are the various chemical names for DDT. And if you could see the history of DDT, this is the first of the modern insecticide, which comes under organochlorine. It is synthesized in the year 1874. Insecticidal activity was recognized in the year 1939, and it gets produced in higher amount in millions, but nowadays people are not using DDT as not only kills the pests, but it is also causing harm to several organisms. And coming to the physical characteristics of DDT, it's a mixture of isomers, up to 30% of OP isomer, and waxy solid of indefinite melting point. And it is white crystalline solid, and its melting point is 108.5 degrees Celsius. And it is soluble in most aromatic and fluorinated solvents. There are different types of analogs are present for DDT. Examples are DDD, DE, and dicofol, methoxychlor. These are the structures of DDT and their analogs. There are commonly used organochlorines are listed here, which these structures already seen previously in the previous slides. These are the banned pesticides because not only it is killing the unwanted organisms or the enemies, but they are also causing harm to the other beneficial organisms too. And coming to the toxic symptoms, it causes cough, shortness of breath, vomiting, diarrhea, etc. We can also treat the symptoms using lorazepam and activated charcoal. Coming to the next point, organophosphate. These organophosphorus compounds are organic derivatives of phosphorus that are largely being used as pesticides and nerve agents, several of which are highly toxic. Organophosphate pesticides are used in commercial agriculture to control pests on fruit and vegetable crops. They are also used in home gardens, mostly control on pests, and in some no pest threats. The main mechanism is blocking the enzyme acetylcholine esterase, causing nervous and respiratory damages that result in the insect death, but they are also hazardous to humans. Coming to the history, in the year 1837, Hoffman synthesized methyl phosphate chloride as an OP compound. Clermont produced tetraethyl pyrophosphate in the year 1854. Although tetraethyl pyrophosphate was not the first synthesized OP organophosphate, it was the first OP cholinesterase inhibitor. In the year 1934, Dr. Gerhard Schrader, a German chemist at IG Farbenenstein, synthesized hundreds of OPs, organophosphates, including parathion as a pesticide, and tabun, sarin, and so many. These are the things. After World War II, American companies began synthesizing organophosphate pesticides in large quantities. Parathion was among the first marketed, followed by malathion and azine phosmethyl. The popularity of these insecticides increased after many of the organochlorine insecticides, such as DDT, Dieldrin, and heptachlor, were banned in the year 1970s because of their long life persistence in the environment. Coming to the mode of action, these organophosphates interfere with the enzymes acetylcholinesterase and other cholinesterases, disrupting nerve impulses and killing the insect. Normally, nerve cells in the brains or muscles of humans or insects send electrical pulses down dendril to the end of the cell, where the pulse has to jump across the gap. Just now we have seen the picture when the insecticide is present, how the gates were open. When the insecticide is not present, it got blocked. Okay, known as a synapse to another nerve cell. A chemical is known as ACH, that is acetylcholine, moves from one cell to the other and binds with the new cell, sending the electric pulse down the new cell. The insecticides and nerve gas agents that are closely related prevent acetylcholine from coming loose from the new cell, so it can't receive any more impulsion. The insect can't function and finally will die. This is the effect of organophosphate on acetylcholinesterase enzyme. Okay, if you could see here, there is a signaling of acetylcholine, and here you are seeing acetylcholinesterase enzyme is stopping signaling process, and here you are seeing the effect of organophosphates on acetylcholinesterase. These are the structures, and these are the commonly used organophosphates: parathion, malathion, solopyriphos, phosmet, etc. And there are several organophosphates were banned. These are the things: chlorpyrifos, diazinon, phenitothioin, parathion, monoxotophos. If you could see the toxicity symptoms, it causes hypotension, cough, nausea, vomiting, pain, abdominal pain, diarrhea, etc. Coming to the treatment, like the organochlorines, here also activated charcoal is used to treat, and breathing support will be provided. Along with that, atropine is used as an antidote. And the last one is carbamate. Coming to the carbamates, these are also insecticides, which are organic compounds derived from carbamic acid. They are more degradable than organophosphates. They have lower dermal toxicities. Coming to the history of carbamate, it is the first successful carbamide, the first successful carbamate, which was introduced in the year 1956. First, it has very low mammalian oral or dermal toxicity. Secondly, it has a rather broad spectrum of insect control. Coming to the mode of action, they have similar properties like organophosphates, which we have studied just now, but last in the environment for a much shorter time period. They are thought to be less toxic. These are the structures of carbamates, different types of the carbamates. These are the different types of the commonly used carbamates. This is the aletharbitha carbamate that was banned. Coming to the mode of action of carbamate. Coming to the mode of action of carbamate, I'll try to explain it in a little different way. These carbamate insecticides enters the synapse and it competes with the acetylcholine, as we have studied in organophosphate stage, which is the active site on the enzyme. This insecticide binds to the enzyme and forms a complex. This complex is sometimes reversible and the enzyme could be disassociated from the carbamate. And here, whatever the things which we have studied for organophosphate, the similar things are applicable here also. Coming to the symptoms of toxicity, you're going to see the symptoms of toxicity in mammals along with the insects. I already told you. Here, under organophosphates, we have studied the treatment, that is the therapy. The antidote for carbamate poisoning is atropine. Here also, for the organic phosphates, it is atropine. Carbamates and organophosphates, its effect and all more or less same. You just read in both the things comparatively. Maximum similarities will be there, except the names, but the function and its effects, everything is same. This is all about the carbamates. So by this we completed the botanical insecticides, organofluorines, organophosphates, and carbamates. This is all about block 2. And now we are going to talk about the other one, which is called as synthetic pyrethroids. One minute, please. I'm sharing the screen. Can you see the screen?
Yes, ma'am.
Yes, madam.
Yes, ma'am. Okay. So now we are going to talk about pyrethroids. This is under block 3, newer insecticides. Hope you understand the things what we have studied till now. And coming to the newer insecticides, we have 3 units under here. The first one is synthetic pyrethroids. Second, and the next unit is molecules with different chemistry. And the third one is synthetic insect control agents. And now we are going to talk about pyrethroids. Yesterday I said that I am going to show you the pictures of neonicotinoids, which I didn't show you in my previous slide, yesterday's slide. I'll show you at the end of this topic or tomorrow if we have time. I'll try to complete this topic at least half. Okay?
Okay, madam.
1 minute, please. I couldn't see this screen. 1 minute. Okay. Can you all see the screen? Is it clear?
Yes, ma'am. Clear.
Yes, ma'am. Clear only.
Thank you. Coming to the pyrethroids, it is an organic compound similar to natural pyrethrins. Whatever we have studied, the pyrethrum under botanical insecticide. Okay, it is similar to this, which is obtained from dried and powdered flowers, daisy like plant, chrysanthemum. Hope you can recollect the things what we have studied today in our previous document. Okay.
Yes,
The name pyrethroids or pyrethrins is given to the active insecticidal compound of a dried flower. It's a combination of 2 carboxylic acid and 3 keto-alcohols. And see, here you can see the carboxylic acid and a keto group. Okay. And they are grown in Kenya and other countries. Recently, Australia has become the world's second largest producer after Kenya. And coming to the history of pyrethrin, French soldiers used crushed flowers to control fleas and body lice during the Napoleonic Wars. We already read these things under pyrethroids. From the year 1803 to 1815. And in the year 1860, pyrethrum powder was introduced to the United States. And in the year 1881, pyrethrum cultivation was introduced to Japan from England, about 82%. And in the year 1914, Japan became the principal supplier of pyrethrum flowers to the United States. And in the year 1970, the US Navy made the first pyrethrum extract against houseflies and mosquitoes. In the year 1949, first synthetic pyrethroids. Our topic is synthetic pyrethroids. So here, in the year 1949, first synthetic pyrethroids, electrin and bioelectrin developed in America by Milton S. Fletcher and colleagues. 20 times more it is 20 times more effective than DDT. It is more active than the natural pyrethrum, but are unstable in sunlight. Pyrethrum is a natural insecticide and pyrethroids are synthetic. In the year 1974, the Rothamsted team have discovered more persistent compounds, permethrin, cypermethrin, and deltamethrin. They are more resistant to degradation by light and air, suitable for use in agriculture, significantly low mammalian toxicities. And coming to the properties of the pyrethroids, it shows a direct contact with the insects to show its attack on nervous system, and its effect is almost immediate. And it degraded by sunlight and air, so they have less environmental hazards. And it's a powerful insect repellent and have knockdown effect too. One of the most least toxic domestic insecticide. It shows low toxicity towards the mammals. And coming to the other properties, it is pale yellow viscous soils and tannous. It is insoluble in water, soluble in organic solvents. In light and air, oxidation and loss of insecticidal activity occur. Hydrolyzed in the presence of alkalis, not readily washed off from plants by rain due to lipolytic character. Coming to the natural insecticides, the chemicals that are obtained from natural sources and used to control insect pests. Pyrethrum, also called as natural pyrethrum, natural pyrethroids, or simply pyrethrin. Highly effective botanical insecticide used for centuries against all manner of insect pests. It occurs in the daisy. This is the scientific name, Thanastratum cinereifolium. Found in tiny oil-containing glands on the surface of seed phase in tightly packed flower heads. Insecticidal activity of this one was discovered in Iran around 1800. It is made up of 6 complex organic esters formed by a combination of carboxylic and keto groups. This we have studied when we are talking about pyrethrum under botanical insecticides. Okay, pyrethrin 1 contains chrysanthemate acid, pyrethrin 2 contains pyrethrin. Okay, this is the composition of these things. And coming to the extraction of pyrethrum, flowers are harvested after blooming, either dried and powdered or the oils are extracted with solvent. Result in pyrethrum usually have an active ingredient content of about 30%. It's an active insecticidal compounds which are collectively known as pyrethrins. This is the cultivation. This is the mortar pistol will be used to collect the things. Coming to the mode of action, it shows its effects on nervous system, and we already studied how the nerve impulses will be and how the information will be passed, how the messages will be passed, how the ion channels works and all. Here, as it is acting on the nervous system, it leads to the paralysis. Okay. And finally, sometimes it may eventually lead to the death of the organism also. Sometimes these things mixed with pyrethroids mixed with other chemical known as synergist. And here the chemical used show their effect in a different way. Either sometimes it leads to detoxification, it leads to increase in the toxicity, etc. Coming to the applications, human lice, mosquitoes, tortoises, beetles, flies, and mites, all these things can be controlled using pyrethroid. And it is highly toxic to cats. Coming to the environmental effects, it is low in toxicity to people and other mammals. It is toxic to beneficial insects such as bees, dragonflies, mayflies, and gadflies. It is very highly toxic to fish, lobster, shrimp, etc. Agent Orange hair rapidly broken down into inactive products and are removed from the body. Irritation of the respiratory passages, running nose, coughing, difficulty breathing, vomiting, and diarrhea. All these are the symptoms. Coming to the different types of the pyrethrum which are available in the market. This is the list. And coming to the synthetic insecticides, originally pyrethroids were made to synthetically mimic pyrethrin, which are derived from naturally occurring pyrethrins taken from pyrethrum of dried chrysanthemum flowers. Chemically more complicated molecules and making more toxic, 10 times more effective than natural pyrethrins. Today's pyrethroids have very low or no odor at all, last longer, can be used in lower concentrations. We already studied about the synergies in the previous slide. Pyrethroids are grouped into 2 classes, type 1 and type 2. Type 1 pyrethroids are the derivatives of pyrethrin, which do not contain cyano groups, but the type 2 pyrethroids contain cyano groups. There are different types of the pyrethrins, which are going to see here. The first one is alethrin, second one is resmethrin, permethrin, phenolphthalein, etc. Coming to the first one, alethrin, it is the first synthetic pyrethroid, which is pale yellow liquid in color. This is the structure of alethrin, which is similar to the top. It shows the action similar to the top of the pyrethroid. It shows contact with the insecticide and shows its effect on the nervous system. It is effective against mosquitoes, flies, and ants. And this alethrin is available in the form of oils, mass, oil formulations, and as an aerosol spray. And coming to the resmethrin, it is a colorless vacuum solid. It is a similar function which we have studied for alethrin. It is not toxic to plants. It is used to control agricultural, horticultural, and public health insect pests. Coming to the permethrin, it is discovered in the year 1973. It is used as a medication to treat fleas, termites, and lice. And it is applied to the skin as a cream or lotion. It is more effective at low temperature. And it is white in color, odorless, crystalline solid, and contact insecticide. Phenolphthalein. It is also called sumithrin and diphenolphthalein. It kills adult fleas, ticks, bedbugs, etc. It is extremely toxic to bees and aquatic life. It shows its effect on insects' central and peripheral nervous system. It is pale yellow color. Coming to the bifenthrin, it is primarily against the red important fire ant. It shows its effect against different types of arthropods or the insects. It is poorly soluble in water and often remains in soil. White wax is solid with a faint sweet smell. It's not naturally occurring insecticide. Steflutrin, it is introduced in the year 1986, which is a colorless solid at room temperature, which is less soluble in water. It shows less flammability. It is effective against soil pests and crops, such as maize, corn, and grains. Tetramethrin, which is a potent synthetic insecticide, it is a white crystalline solid, degrade rapidly after it applied. It is metabolized quickly. It shows its effective action against different types of insects like ants, mosquitoes, roaches, hornets, wasps. Cycloprin, which shows its effect against different types of insects like cutworms and silverfish, etc. It is available in the form of emulsifiable concentrates, sweatable powder, aerosol, granules, liquid. Yesterday we have studied about the formulations and all. At that time we have covered these things. It shows both contact and stomach poison action. Cyhacloprin, it shows effective for longer periods of time. Colorless solid with a mild odor. It shows low water solubility and it is non-volatile. It forms strong bonds with soil, so no cross water contamination. It is effective against aphids, Colorado beetles, and butterfly larvae, mainly used as a picticide. Coming to the next one, cypermethrin. It is popular and widely used insecticide, stomach and contact insecticide, behaves as a fast-acting neurotoxin in insects. It is easily degraded on soil and plants, but effective for weeds when applied to indoor inert surfaces. It is effective against cockroach, flies, mosquitoes, etc. And deltamethrin, it is most popular and widely used insecticide. It is colorless crystalline powder. It shows its effect against different pests on different pests of fruits, vegetables, pests of fruits, vegetables, cotton, and stored products. It controls spiders, bedbugs, ants, etc. It plays a key role in controlling malaria vectors, particularly Anopheles gambia. Fenvalerate, the same functions, similar to the of the things which we have studied till now, contact and stomach poison. Yellow brown viscous liquid practically soluble in water, commonly used to control insects in food, fish, cotton products, etc. Does not affect plants. Fenflupathrin, it is an insecticide as well as an acaricide. It is used to control aphids, red spiders, etc. It is risk for Parkinson's disease. Flucetrinate, it is an insecticide, and acaricide, it is highly toxic pesticide. Currently, it is not approved for use in many countries along with the United States. And it is effective against phytophagous mites and insects of cotton and vegetables. Flumethrin, it is externally used in veterinary medicine against parasitic insects such as flea and ticks. It is used in the treatment of parasitic mites in honeybee colonies. Fluvalinate, it is viscoscellulose oil. It is stable, non-volatile, fat-soluble compound. Acaricide, it is also an acting as acaricide, commonly used to control varromites. It shows its action against moths, beetles, and other insect pests on cotton, cereal, grape, potato, fruit tree, vegetable, and ornamental plants. Coming to the merits of pyrethroids, they are extremely high insecticide. They show extremely high insecticidal activity at extremely lower doses. Biodegradable in nature, it is effective against lepidopterous pests and beetles, leaf miners, and bugs. They are effective against eggs, larval, and adult stages of insects. Powerful antioxidant and repellent properties. No readily washed off from the plant by rain. Movement from site of application is limited in air due to low volatility. Coming to the demerits, not systemic, and do not have translaminar action. Constant use of synthetic pyrethroids increase resistance in insect pests. Harmful for beneficial organisms like honeybees, beetles, etc. They are highly toxic to fish. On prolonged storage, pyrethrum loses its insecticidal action. Hence, antioxidants like hydroquinone are added. So this is all about your pyrethroids, different types of the pyrethroids, and the function of pyrethroids, where they are available, how they are available, what is its composition, what is the mode of action. By this, we completed pyrethroids, synthetic pyrethroids. Whatever the things which we have studied, all these are the synthetic pyrethroids. In our next slide, we are going to talk about the molecules with different chemistries. Hope you
Excuse me, madam.
understand. Amma,
What is meant by translaminar action?
translaminar
Action.
related to
Pyrethroids. The last topic which we have discussed now.
okay, it is an insect. Normally, it is an insecticide, as we all know that pyrethroid is an insecticide. Which contains different types of ingredients, chemical components, which helps to control the pests.
Okay.
Okay.
Okay, madam. Madam, please share this PPT and PDFs in the group, no, madam.
Yes, we'll share. I'll share. Once I'll align everything, once I'll complete each block, I'll share the PPTs and all.
Okay, madam. Thank you.
Okay. Clear?
Yes, madam. Clear.
Okay. If you have any doubts, ask me now. Shall I close? Shall we close for the day? Hello?
Yes, madam.
Yes, we'll close for the day. We'll join tomorrow to study about all these things. Once we complete all the topics at once, we'll go for the revision, where we are going to talk about the important topics, important points and all. Okay. First, we'll finish it off with all the things.
Okay, madam.
Okay. Thank you all. Bye.
Thank you, madam.